So this is a review video of the light reactions of photosynthesis. This is the beautiful schematic that I will be using for most of the video. This represents the thylakoid membrane. Remember, it is a phospholipid membrane. I just didn't draw the phospholipids. Thus, this is going to represent the inside of the thylakoid throughout the duration of the video, and the upper half is going to represent the stroma, or the fluid that surrounds the thylakoid. These represent the molecules within the thylakoid membranes. You've got your photosystem 2 and the molecules that are associated with it, and photosystem 1 and the molecules that are associated with it. So the first step of the light reactions is that light from the sun is absorbed by photosystem 2. Now remember, a photosystem is just a group of pigments, both primary and accessory, that is located within the thylakoid membrane. And as light strikes the photosystem, energy is passed between the molecules until it reaches a specific pair of chlorophyll A molecules. The chlorophyll A, remember, is located within photosystem 2. So the light energy that is transferred to the chlorophyll A molecules forces the electrons to enter a higher energy level, which actually gives them enough energy, they're excited, to leave chlorophyll A. And since the electrons will be leaving the molecule chlorophyll A is losing electrons, it's losing negative charges, it's becoming more positive, so chlorophyll A is oxidized. Where are those electrons going, you ask? Those electrons are going to the primary electron acceptor of photosystem 2. Since the primary electron acceptor is gaining electrons, its charge is becoming more negative, and the primary electron acceptor is said to be reduced. Remember, those electrons came from the excited electrons of chlorophyll A in photosystem 2. So what happens to the electrons after they make it to the primary electron acceptor of photosystem 2? They then move through the electron transport chain of photosystem 2. Now, the electron transport chain is just a series of molecules in the thylakoid membrane that transfers electrons from molecule to molecule. As the electrons move through the electron transport chain, they lose energy. Since energy cannot be created or destroyed, that energy has to be used for something. And the energy that the electrons lose as they are transferred between molecule to molecule of the electron transport chain are used to pump hydrogen ions into the thylakoid. Notice that hydrogen ions have a positive charge, which means you're building up positive charges within your thylakoid membrane. And remember, the energy to pump those hydrogen in comes from the energy the electrons lose as they move from molecule to molecule within the electron transport chain. And it requires energy, so it's active transport, just so you remember. While this is going on, at the same time, light from the sun is also hitting and being absorbed by photosystem 1. And just as in photosystem 2, that light, which once again possesses energy, is being transferred from molecule to molecule within photosystem 1 until it reaches a specific pair of chlorophyll A molecules, which hopefully sounds familiar because that's the same thing that happened in photosystem 2. These electrons become excited, and once again they have enough energy to be transferred to the primary electron acceptor of photosystem 1. Once again, the primary electron acceptor is gaining electrons, so it's being reduced, and the chlorophyll A molecules that lost them were being oxidized. So what happens in photosystem 1 after the electrons have been transferred to the primary electron acceptor? They then go through the electron transport chain of photosystem 1. And the electrons in photosystem 1, instead of being used to pump hydrogen into the thylakoid, the electrons are actually brought to the stroma side of the thylakoid membranes, and the electrons of the that pass through the electron transport chain are actually donated to the stroma of the chloroplast. So after your light reactions, you have a bunch of electrons on the stroma side, and even though they're not shown, you have a bunch of hydrogen ions on inside your thylakoid. So you have an electrical gradient because you've got negative charges here and a bunch of positive charges here. And that electrical gradient is really important, as you will see when we talk about chemiosmosis at the end of this video. Now, these electrons are actually used to combine with NADP+ and hydrogen ions in the stroma to give you NADPH. And remember, NADPH, like ATP, is a type of molecule that is used to store energy. And the NADPH that is created here is important because it's used in the Calvin cycle. Used in the Calvin cycle, super important. So the electrons from photosystem 1 are used in the stroma to produce NADPH, and that NADPH will then be used by the Calvin cycle. So how are the electrons lost by chlorophyll A in both the photosystems replaced? Because if they're not replaced, then photosynthesis would have to stop. And it happens in two different ways for the different photosystems. So the electrons in chlorophyll A of photosystem 2 are replaced by the splitting of water. So you have your water molecule. Remember, a water molecule is one of the reactants of photosynthesis. It splits to give you 4 electrons, and those electrons are used to replace the electrons lost by the chlorophyll A molecules. It also produces hydrogen, which adds to the hydrogen concentration inside the thylakoid, and it produces oxygen, which then diffuses out of the plant, and that's how we get oxygen from photosynthesis. But water is the electron donor, and it replaces the electrons in photosystem 2. In photosystem 1, the electrons are replaced by the electrons that have passed through photosystem 2. So the chlorophyll A in photosystem 1 gets its electrons from those that have passed through photosystem 2. And then all your electrons are replaced, and photosynthesis can continue to happen over and over and over and over again. At the end of that, as noted before, you have a bunch of electrons in here, and then you have a bunch of positive charges inside the thylakoid, and as stated before, that creates this electrical gradient. And you might be asking yourself, why is that electrical gradient important? And that electrical gradient is important because it is the key for ATP to be produced at the end of the light reactions through the process chemiosmosis. So in chemiosmosis, what happens is, because you have this high concentration of hydrogen ions in the thylakoid, inside the thylakoid, and remember, diffusion tells us that molecules generally want to move from areas of high concentration to areas of lower concentration. And when they do that, it actually releases energy. And here we have this super high concentration of hydrogen ions. Some we get because they're pumped via the processes that happen in this area from photosystem 2. And some of them get there, remember, from the splitting of water. And both of those contribute to the high concentration of hydrogen ions inside the thylakoid. And like I said, when atoms, molecules, or ions, in this case we're talking about ions, move from areas of higher concentration to lower concentration, energy is released. In this case, we have a molecule that spans the thylakoid membrane, just like all your other molecules, and as the hydrogen goes through that molecule, the ATP synthase, and goes from an area of higher concentration to an area of lower concentration, ATP synthase harnesses that energy, and it uses it to create ATP by adding a phosphate group to ADP. And that ATP is then used in the Calvin cycle. So once again, as hydrogen move through ATP synthase, they're moving down their concentration gradient, which releases energy, and that energy is used to add a phosphate group to ADP in order to create ATP, which will then be used in the Calvin cycle. Hopefully this video is helpful. Good luck on your test.